Hello, my name is Michelle Lee, and I am here today to give you a mini lesson on the challenges that we face when we try to develop a vaccine for a virus. Really relevant topic. Currently, time of recording, this is June of 2020, and we are very much in the midst of the COVID-19 pandemic, so this is a very relevant topic to be discussing right now. There's three challenges that we're going to discuss briefly here. First one is that viruses are easily mutated, and so they turn into something a little bit different, makes it harder for the body to recognize. Second is time that it takes to develop a vaccine. And number three, the money it takes to develop a vaccine. So let's take a look at this first challenge, which says, which explores the idea that viruses mutate really, really quickly. Vaccines are very specific. When they're developed, they're very specific to the antigen that each virus presents to the body. And since viruses are made up of what drives them is single-stranded RNA, that makes that nucleic acid very vulnerable to mutations. And the problem is if the RNA of viruses gets mutated easily, then that potentially changes the antigens on the surface of the virus, making the antibody response that the body has developed to the first antigen is going to be very, very tricky for the body to recognize the mutated version. So the vaccines become much less effective after a virus has mutated, making them unrecognizable, and then the body is left defenseless. I'm going to share with you right now a video that is produced by the California Academy of Sciences on virus mutation.
We usually think of evolution happening very slowly, over billions of years. But bacteria and viruses can evolve in much less than a human lifetime, and scientists have to keep up. Consider H1N1, the virus linked to swine flu. Swine flu has actually been around for a long time, but the virus that causes it can transform very quickly.
Viruses are probably as old as life, and it's quite likely that there will be new lineages of viruses emerging as we go forward in time. So they're very old and they're very new simultaneously.
Very old and very new? That's because ancient virus lineages can mutate, that is, change their genetic structure very rapidly.
It actually has a high rate of mutation. And actually, to properly understand kind of the evolution of the influenza virus, it's important to understand the mechanisms by which the virus can mutate or change. Actually, influenza also consists of 8 different segments. And actually, if you have a mixture of the segments, for instance, if you have some segments that come from pigs, some segments that come from birds, some segments that come from humans, you can then produce an entirely new virus. That's called antigenic shift. And that actually is the cause of a new virus causing a pandemic. All of the pandemics in the past century, the 1918 Spanish flu, the 1967 Hong Kong flu epidemic, were because of antigenic shift changes in the genome. On the other hand, there's another method of change, and that's called antigenic drift. Antigenic drift occurs when you have very small changes in the actual sequence of a currently circulating strain. And this is the cause of what we call seasonal influenza. This is the reason why people can become infected by influenza year after year after year. Despite having had influenza before, you could very easily get influenza again.
These rapid changes keep health officials on their toes. What happened recently may be an antigenic shift event. The virus looks like an entirely new variant. But the problem could get worse because of antigenic drift.
By the time autumn rolls around, when you start seeing the next season of influenza, there may be subtle, very small changes in the genome that will affect the transmissibility, the virulence of this virus, whether or not it's going to cause more severe disease, whether or not you're going to see it again.
So the second challenge that we can talk about when developing a vaccine is the amount of time that it takes to develop that vaccine. It's a methodical, intentional process to ensure that you have a safe yet effective vaccine. And there's a number of stages that are involved in this. So first, scientists have to explore they have to figure out what the genetic structure of the virus is, and to try to figure out what is the proper antigen that they can introduce in with the vaccine. Then once they feel like they have a vaccine that's going to work, they'll bring it to preclinical trials, which will include animal testing. This is followed by clinical trials, which in an ideal world has 3 stages, starting with small groups of people testing while minimizing the possible side effects and damage that can be done. Then they'll introduce the vaccines to more targeted groups. These are the people who are most likely to contract the virus, and so they'll test that population, and then they'll do more widespread use of that vaccine and still kind of follow up on it. This is followed by regulatory review and approval, making sure that this is all being done while following all the laws and to ensure public safety. And then there's just the manufacture and the quality control. So this is typically a timeline that takes quite a bit of time. Traditionally, it will take, you might have some clinical trials going on. That's the phase 1, phase 2, phase 3, and then manufacturing and large-scale manufacturing and all the licensure is taking quite a bit of time. But when you have an outbreak and, like the pandemic, which we're currently experiencing, a lot of things have to be kind of doubled up. You can see here that manufacturing is kind of happening much earlier in the game, really before you're really ready. So everything is really condensed. We're trying to make it faster, but it still takes time. So this next video I'm going to show you is from the Jackson Lab, and it's going to talk to you a little bit about how we can accelerate the development of vaccines.
Traditional vaccine development is slow by design, with extensive scientific review, rigorous preclinical testing, and at least 3 phases of progressive clinical trials. Historically, it takes between 5 and 10 years and over $1 billion to develop a vaccine and to make sure it's safe and effective, and that's before making enough vaccine for everyone and administering it to patients around the world. So how do vaccines work, and how can we speed up the process? When a virus like SARS-CoV-2 enters your body, your immune system responds by producing specific antibodies to both neutralize the current infection and watch out for that virus's characteristic pattern should it ever reappear. Traditional vaccines use all or part of a killed or weakened virus to teach your immune system to recognize and act on those specific characteristics without the virus ever entering the body. Because these vaccines start with live pathogen, they require extensive biosafety facilities and significant time and cost to develop. New vaccines are taking a different approach. Using advanced sequencing technologies, scientists are able to obtain and analyze the genetic code of a new virus within hours of discovering it. After isolating the code for the virus's specific and recognizable proteins, scientists can create a template for that protein, a short, single-stranded sequence of nucleotides called messenger RNA. If the mRNA sequence can be delivered to enough cells, they will create the viral proteins right within the cell, giving your immune system the means to recognize and fight any virus with these characteristics. RNA vaccines can be made in large quantities in a matter of days at relatively low cost. However, they expire quickly and must be stored at extremely low temperatures, making distribution difficult. Despite these challenges and the need for more testing, RNA and other novel vaccine methods have the potential to significantly accelerate vaccine development, and by refining the process for preclinical testing and clinical trials, we will soon be much more agile in the fight against infectious disease.
The third challenge facing the development of a vaccine is the money that it takes to bring a vaccine not only from the research and development phase into a manufacturing phase. So, even just the simple manufacturing costs of the raw materials, maintenance and labor, to local and international law compliance. Funding currently typically comes from national governments and philanthropic groups, but in the end, it's not that profitable for pharmaceutical companies to create vaccines because there's usually just one-time treatments. And so there's a little bit of a profit pressure that doesn't really motivate big companies from wanting to do this. So that's definitely a third and final challenge. Not going to spend much more time on that because this is intended for a high school biology audience. So what makes an antiviral vaccine so challenging to develop? Just to kind of close this out, it comes down to three simple things. Viruses easily mutate, making them difficult to target consistently. Time it takes to develop the vaccine traditionally has been decades. In a pandemic situation, we can kind of shorten it up to within a year or two, but it still takes time, and a lot of lives are in jeopardy and have been lost as a result of that time. Third challenge is money. It takes money to make these things happen. Anyway, hope that this gave you an insight into vaccines and how to combat viruses, especially in this age of pandemics. And I hope this video also gave you a chance to see me, Michelle Lee, as a teacher in action. Thank you for your time.